Welcome to the PubMed Daily Review for March 20 SD. Today we scanned 5,878 new open access papers. Here are the top 4 selected for detailed analysis. Up next, our first paper out of 4, a foundation model for nucleotide sequences by Shilin Shun and Xiangchun Li. Let's break down its key contributions. Foundation models are revolutionizing AI, but in genomics, we've largely focused on raw sequences. However, biological annotations hold vital semantic context. This paper introduces OmniNA, a novel foundation model designed to learn from both nucleotide sequences and their associated annotations, aiming to unlock new levels of understanding in genomics and transcriptomics. The researchers developed OmniNA, a large-scale foundation model built on the Llama architecture. It was pre-trained using a self-supervised approach on a massive dataset of over 91 million nucleotide sequences and their associated annotations. The model's core innovation lies in its ability to jointly learn from both sequence syntax and annotation semantics, paving the way for more comprehensive genomic understanding. The results are impressive. OmniNA, after fine-tuning, achieved state-of-the-art or competitive performance across 23 different benchmarks. This includes tasks like sequence detection and species classification. Furthermore, its learned representations proved valuable in revealing mutation effects on DNA and RNA processing, showcasing its broad applicability. This study marks a crucial step towards more powerful foundation models in genomics. By jointly learning from sequences and annotations, OmniNA offers a versatile tool that can accelerate research and discovery. Its ability to generalize across tasks suggests exciting potential for future applications, from understanding disease mechanisms to developing novel diagnostic tools. Up next, our second paper out of 4, Efficacy and Safety of Psilocybin in Treatment-Resistant Major Depression. The episode, Randomized Clinical Trial by Leah J. Mertens and Gerhard Grunder from Department of Molecular Neuroimaging, Central Institute of Mental Health, Medical Faculty Mannheim, Heidelberg University, Mannheim, Germany. Let's break down its key contributions. Major depressive disorder is a widespread condition, and for many, existing treatments don't provide relief. This leads to treatment-resistant depression, or TRD, which has a profound impact on individuals and society. We desperately need new therapeutic approaches for these patients. This was a Phase 2b, triple-blind, randomized clinical trial conducted at 2 centers. They enrolled 144 adults diagnosed with treatment-resistant depression. Participants received either 25 milligrams of psilocybin, 5 milligrams of psilocybin, or an active placebo, nicotinamide, all combined with psychotherapy. The primary goal was to see if psilocybin led to a significant reduction in depressive symptoms after 6 weeks. The study found that at 6 weeks, the response rate for the primary endpoint was 17% for the 25 milligrams psilocybin group compared to about 11% for the placebo group. However, this difference was not statistically significant, with a p-value of 0.19. Importantly, secondary outcomes did show clinically meaningful reductions in depressive symptoms for those who received the 25 milligrams dose. The treatment was generally well tolerated, though some adverse events were noted, including increased reports of suicidal ideation on dosing days. So, what does this mean for clinical practice? While the primary goal wasn't met statistically, the study suggests that 25 milligrams of psilocybin, when combined with psychotherapy, might still offer real benefits for patients with treatment-resistant depression. However, we must acknowledge the safety signals observed, particularly around suicidal ideation, which require careful attention. This trial adds to the growing body of evidence, but more research is definitely needed to fully understand psilocybin's role in treating depression. Up next, our third paper out of 4, a distinct tau oligomer strain defines the molecular and proteomic landscape of rapidly progressive Alzheimer's disease by Tayaba Salim and Ingezair from Neurology, University Medical Center Göttingen, Robert Kochstrasse 40, 37075 Göttingen, Germany. Let's break down its key contributions. Alzheimer's disease, the most common cause of dementia, isn't a one-size-fits-all condition. While many experience a slow decline, a rare, aggressive form called rapidly progressive AD, or RPD, leads to swift cognitive deterioration. We don't fully understand what makes RPAD so aggressive at a molecular level, especially concerning tau protein, a key player in AD pathology. This study aimed to investigate if specific forms of tau oligomers could be the key to understanding these differences. To tackle this question, the researchers focused on tau oligomers, or tau-O, which are thought to be particularly toxic in AD. They isolated these tau-O from the brain tissue of individuals with normal cognition, slow AD, and the rapid form of AD. Using advanced techniques like electron microscopy and mass spectrometry, they analyzed the structure, protein interactions, and toxicity of these tau-O. They also used a mouse model to see how these human tau strains behave in a living system. The results were striking. Tau oligomers from RPAD brains showed higher levels of specific phosphorylation at PS 396 and PS 422 compared to other groups, reaching statistical significance. Proteomic analysis revealed a core set of proteins associated with tau oligomers, but RPAD had a significantly larger unique set of interacting proteins. These unique RPAD proteins were linked to processes like aldehyde detoxification and metabolism, rather than the proteostasis and mitochondrial pathways seen in controls and slow AD. Importantly, tau oligomers from both SPAD and RPAD, but not controls, reduced neuronal cell viability in lab assays. So what does this all mean? This study strongly suggests that rapidly progressive Alzheimer's disease is driven by a specific, biochemically unique strain of tau oligomers. These RPAD tau are not just different in structure, but are also part of a distinct molecular network, particularly involving metabolism and the cell's internal scaffolding. Understanding these differences opens the door to developing targeted therapies specifically for RPAD, potentially offering new hope for patients with this aggressive form of the disease. Up next, our fourth paper out of 4. Transcutaneous spinal cord stimulation plus locomotor training versus sham stimulation plus locomotor training in chronic spinal cord injury. EWalk, a multi-center, triple-blind, randomized, sham-controlled trial by James B. Krashia and John D. Steve from University of New South Wales. Let's break down its key contributions. Spinal cord injury can severely impact a person's ability to walk, making regaining this function a top priority. While locomotor training is a standard rehabilitation method, its effectiveness can be limited. There's a growing interest in combining different approaches for better outcomes, and transcutaneous spinal cord stimulation, or TSCS, alongside locomotor training has shown some promising early results. This was a multi-center, triple-blind, randomized sham-controlled trial called EWalk. They enrolled 50 individuals with chronic spinal cord injuries who had limited walking ability. Participants received either active transcutaneous spinal cord stimulation or a sham stimulation, both combined with 12 weeks of locomotor training. The primary goal was to see if the active stimulation group showed greater improvement in walking ability compared to the sham group. The study found that after 12 weeks, the group receiving active TSCS showed a mean difference of just -0.1 points on the WSI-2 walking scale compared to the sham group. This difference was not statistically significant, with a p-value of 0.98, and, importantly, it did not meet the predefined clinically meaningful difference of 2 points. Both groups did show improvements in walking, which were maintained at follow-up, and adverse events were similar between groups, with no serious events reported. So, what does this mean for clinical practice? This well-conducted trial suggests that this particular approach of using TSCS alongside locomotor training isn't more effective than locomotor training by itself for improving walking in people with chronic spinal cord injuries. While this specific protocol didn't show benefits, it opens the door for future research to investigate TSCS in other SCI populations or with different stimulation and training strategies.